Welcome back to Acing AP Biology. In this lesson, we will be discussing the process of fermentation. When oxygen is not available, the pyruvate molecules from glycolysis cannot enter the Krebs cycle. So instead, fermentation takes place. Fermentation is the anaerobic catabolism of organic compounds to produce ATP. The purpose of fermentation is to regenerate NAD+ so glycolysis can continue. It is important to remember that fermentation does not produce ATP, but regenerates NAD+ so glycolysis can continue to produce ATP through substrate-level phosphorylation. Compared to cell respiration, fermentation produces 18 times less ATP. There are two major types of fermentation: lactic acid fermentation and alcohol fermentation. The process of fermentation starts with glycolysis, where glucose is partially oxidized to form two pyruvate molecules. 2 ATP and 2 NADH molecules are produced in this process. If oxygen is available, Krebs cycle and oxidative phosphorylation will ensue. If oxygen is not available, fermentation will take place in order to regenerate NAD+ for glycolysis to continue, so ATP can continue to be produced. In lactic acid fermentation, pyruvate is reduced to lactate, and NADH is oxidized into NAD+. Therefore, the NAD+ is regenerated for glycolysis. In alcohol fermentation, pyruvate is converted to a C2 compound called acetaldehyde, and a CO2 molecule is lost in this process. Then acetaldehyde is reduced to form ethanol, and NADH is oxidized to NAD+. As I mentioned previously, alcohol fermentation begins with glycolysis, resulting in the production of 2 pyruvate molecules. Now, since oxygen is not present, fermentation takes place. The pyruvate molecules are converted to acetaldehyde with the loss of carbon dioxide. Then, the acetaldehyde is reduced to ethanol, and NADH gives up its electrons to acetaldehyde, being oxidized to NAD+. Therefore, acetaldehyde is the final electron acceptor in this pathway, and NAD+ is regenerated for glycolysis to continue. Yeast and some bacteria use this process in anaerobic conditions to produce ATP. Next, we have lactic acid fermentation. Lactic acid fermentation also starts with glycolysis, in the formation of 2 pyruvate molecules. NADH donates electrons to the pyruvate molecules, and they are reduced to lactate. NADH is therefore oxidized to NAD+, and the NAD+ goes back to glycolysis. Furthermore, pyruvate acts as the final electron acceptor in this process. Lactic acid fermentation is very important in the human body. Muscle cells switch from aerobic respiration to lactic acid fermentation when oxygen levels in the cells are low, like when you are exercising. As a result, lactate builds up and is then carried to the liver to be converted back to pyruvate once enough oxygen becomes available for aerobic respiration. This is why your muscles feel sore after exercise, due to the lactate buildup. Lactic acid fermentation is also used in the fermentation of dairy products like yogurt and buttermilk. That's it for Unit 7. Cellular respiration is a complicated process, so if you have any questions, feel free to comment below. In the next unit, we will discuss photosynthesis. See you then! For more bio content, hit subscribe.